Good evening. My name is Anita Pepper, and I'm Vice President of Institutional Advancement at the Wistar Institute. I have to say that 2021's gotten off to a rough start for everyone, but one of the things that has kept me positive and forward-thinking is knowing that I get to introduce you all to the inimitable force that is Dr. Bonnie Bassler, and to present her with the 2020 Wistar Institute Helen Dean King Award. I want to start by giving my sincere thanks to the members of the Women in Science Program Committee, the sponsors, and all of you who have generously supported this initiative and Wistar Science. Wistar is an international leader in biomedical research and training and is the home of breakthrough scientific discoveries in cancer, immunology, and infectious disease research. Wistar's mission as an independent research institute is to let our scientists focus solely on the science. We give our researchers financial support and a collaborative environment to make new discoveries in biomedical science and to push those discoveries from the laboratory bench to therapies and solutions that will improve human health. Wistar's Women in Science program highlights the crucial role that basic and clinical research play in addressing the scientific challenges related to women's health and showcases the distinguished and dynamic women behind the science. When science was an entirely male-dominated field, Wistar hired its first female professor, tonight's namesake, Dr. Helen Dean King. She started working at Wistar in 1908, and at that time was the only woman in America to hold a full professor in research. She spent the entirety of her career at Wistar until her retirement in 1950, making seminal contributions to science, such as the development of the laboratory model still used and known today worldwide, the Wistar rat. Like Helen Dean King, when I think of bold, intelligent, and paradigm-shifting scientists, Bonnie Bassler is the top of my list, and it's a great honor for me to present her with the Helen Dean King Award. I would like to share all of Bonnie's many honors, awards, and contributions to science, but that would take up all of our time with a 28-page CV. So I will pick some highlights from her incredible career thus far. Bonnie received a BS in biochemistry from the University of California in Davis and a PhD in biochemistry from the Johns Hopkins University. She performed postdoctoral work in genetics at the Gorin Institute in California and joined the Princeton faculty in 1994, where she is currently a Howard Hughes Medical Institute investigator, the squid professor, and chair of the Department of Molecular Biology. Bonnie is a member of the National Academy of Sciences, the American Academy of Arts and Sciences, the National Academy of Medicine, the American Academy of Microbiology, and the Fellow of the American Association for the Advancement of Science. If that's not enough, Bonnie was also elected to the Royal Society and to the American Philosophical Society, in addition to many more I did not mention. Her commitment to advance science for a diverse workforce is relentless, and her list of committees, scientific advisory boards, and board of directors that she helps guide is unlike any I have ever seen. One highlight is that she was a member of the National Science Board, nominated to that position by President Barack Obama in 2010. Bonnie has received numerous awards for her work, including the Gruber Genetics Prize, the Genetics Society of America Medal, the Shaw Prize, the Schering Prize, the Dixon Prize, the Max Planck Research Award, the UNESCO L'Oreal Women in Science for North America Award, the Lounsbury Award, and MacArthur Fellowship, and I did cut the list a lot. In addition to receiving honorary degrees from 5 universities, she sits on many editorial boards, scientific education committees, and awards panels as well, which is where I had the good fortune of crossing paths with Bonnie now quite a few years ago. Bonnie is most notably recognized for discovering how bacteria communicate with one another in order to orchestrate group behaviors. This process, known as quorum sensing, is best described by Bonnie, but I have no doubt you all will never think of bacteria the same way after this presentation. It was a huge disappointment that we had to postpone this event last year, so it was with great pride that we now present and congratulate Dr. Bonnie Bassler with the Wistar Institute Helen Dean King Award, honoring outstanding women in science for their courage, leadership, and contributions to advance human health. So with that introduction, Bonnie, may I pass to you your Helen Dean King Award?
Thank you, Anita. Oh, thank you. And I want to show, first I wanted to show everybody this beautiful statue of the Wistar rat. I guess everyone can see it.
Yes, it's
right? It's a really fantastic art piece. So Anita, thank you for that very lovely introduction. I am delighted to receive this award because of these heroic women that came before me and pushed the boundaries of science, and those women let women like me do the science that I do. And so I'm really indebted to Dr. King and it's amazing to be on the list of women who have won this prize. And I just want to thank the Wistar for celebrating science, for making it easy to do science, and to make basic discoveries that translate into medicine, and for always having their eye on how women can move through the ranks. It's really just an incredibly meaningful award, and it's been a year in the making. I've had a lot of time to get excited about tonight. So thank you for this introduction. I'm super, super delighted.
Wonderful. And now I'm going to pass the screen over to you, Bonnie, and we get to hear your wonderful talk, Tiny Conspiracies, How Bacteria Talk to Each Other.
Okay. So Anita gave a fantastic introduction into the work that my gang does in Princeton, New Jersey. And so I will give you a whirlwind tour of that. And of course, I'm a teacher, and so I have a goal for this lecture. My goal is that at the end of these 45 or so minutes is that you think that bacteria can talk to each other. And then if I manage to achieve that goal, my secondary goal is to try to help you understand that they're multilingual. And then the third thing I'd love to accomplish tonight is to try to convince you that learning about how bacteria talk to each other, learning their languages and how that works, is giving us insight into bacteria so that we can interfere with communication and hopefully make new kinds of medicine. So that's the arc of the talk. And with that, let me get going. And the first thing I want to do is to just give a little background and make sure that we're all on the same page about what bacteria are. So this first slide shows a cartoon of a typical bacterium that you might, like an E. coli, that you might find in your gut. And most bacteria look pretty much like this. They are single-celled organisms. They are too small to see with the naked eye. You have to look in a microscope to see a bacterium. And they're pretty much built like this guy, which is that they are surrounded by a membrane, and that keeps the outside world out and the inside world in. And inside of them, they have cytoplasm that has all kinds of biomolecules roiling around. And then they all have one chromosome, so one piece of DNA that has all of the genetic information on it that gives them their physical form and then all of their behaviors. And so scientists have known about bacteria for over 500 years. They are Earth's most primitive organism. They're the first living organism on Earth. They've been on Earth for billions of years. And for almost all of these 500 years, they were always thought to live these sort of mundane, reclusive lives. And so what I mean by that is if one looks at them under a microscope, what one would see is that bacteria consume nutrients from their environments. They grow to twice their size. They double all of their components. They cut themselves down the middle, and one cell becomes 2, becomes 4, and so on and so on. So they sort of eat and divide and make progeny. And so because they look so primitive, single cells, and because when you just look at them under a microscope, they don't seem to do many tricks, they've always been thought to be asocial. And what I mean by that is that when a cell divides into 2 sister cells, the sister cells have no knowledge or no interaction with one another. And I want to try to convince you that that is not true. And the fact that they do interact and that they are social is what gives bacteria their power on Earth. And so before I get to that, I want to make sure that you're invested in this talk. And so I thought I would start tonight by just telling you a little bit more about your relationship, your everyday relationship with bacteria. And so these next couple of slides will actually also give you insight into my view of the world. So this is my sort of depiction of a human being. So what I've done is to make this picture of this man, right? And all the little circles represent some of the cells, the human cells that it takes to make a human being. And so there's some trillions of cells, right? That's how many cells it takes, how many cells you have in your body that make a human being. So at any time in your life, you have 10 times more bacterial cells than human cells in you or on you. So if you want to use this first metric that I have here, you are actually only 10% human. You're 90% cells, 90% bacteria, at least by cells. But of course, it's not just the cells, it's the DNA. And so you guys know that the DNA is this magical double helix that's our genetic blueprint that gives us the information that encodes all of the components in our bodies. And of course, you also know that the human genome has been sequenced. So I've remade my man out of the famous A's, G's, C's, and T's that make up the DNA double helix. And so we know about this from the Human Genome Sequencing Project. There's about 25,000 or 30,000 genes that's in the human genome. So it's 30,000 bits of information that give you your physical form and all of your behaviors. So it turns out that you have 100 times more bacterial genes in you or on you than you have human genes. So you can use either of these metrics. At best, you're 10% human. In my view, you're only 1% human, because 99% of the genes that are playing out in your day-to-day life come from bacteria, not from your own genomes. So these bacteria live in us and on us in a magical community that's called the microbiome, and it is essential for life, because the jobs that these genes and these bacterial cells are doing, you can't do. Your own genome doesn't have the capacity to do them. And so now what we understand is that these microbiome bacteria are our 24/7 partners, and they make all of the rest of life on Earth possible. So this is sort of how I look at the world. And so what we're starting to understand now is that bacteria have basically these two personalities. So the historic one, the one that we've known about, or scientists have known about for a couple hundred years, is that there's all kinds of bacteria that live in the world that are pathogens, right? I don't want to pretend there's not harmful bacteria. There's terribly harmful bacteria. And so on the top row, you're seeing pictures under the microscope of notorious pathogens that if these bacteria get in us or on an animal or on a plant, they can make us sick or they can kill us. Right? So there's all these bad bacteria in the world, but increasingly what we're learning about is this microbiome, that there's also all kinds of beneficial bacteria that do these magical jobs that make life for higher organisms possible. So these now are facts. There are bacteria that can kill us, and there are bacteria that are keeping us alive. And so those are facts. We know them from those facts from scientific studies. So that's not actually, so that's just known now. But the part that I'm interested, or that my gang is interested in, is not whether bacteria can be harmful or beneficial. What we want to try to understand is, how can they do either of these things? They're so tiny. So how can it be that these little bitty critters can either kill us or they can give us life itself? And that's what my lab always tries to understand. We try to understand, good or bad, how do bacteria get this power? And so what I want to try to tell you tonight is the way they do these tricks is by communicating with a chemical language, by counting their numbers, and by taking on these tasks as groups. So they act together as collectives, and when they all do something in synchrony, they can actually manage tasks like the ones on this slide that they could never manage if an individual bacterium acted alone, because individually they're too small. And so that's where we're going. I'm going to try to show you bacteria can talk and that they have group behaviors. And so since this is this special talk on this nice evening, I thought what I would do is to just tell you about how we got to this idea of bacteria communicating and working in groups, sort of the history of the field, because it comes from a really quirky initial observation. It actually comes from an observation that was made about this animal, which is called the Hawaiian bobtail squid. So this squid lives off the coast of Hawaii. It's a couple inches big, and it's a nocturnal animal. So during the day, the squid sleeps, and at night, it comes out and it has to hunt for food. All right, so this squid is in an aquarium. I'm going to show you a movie of the squid. So what you're looking at is this squid, and the lights are on. So the squid thinks that it's daytime, and it looks kind of spastic and nervous because, as I told you, it's a nocturnal animal. So in the day, it's not supposed to be out swimming around, because if it was, a predator would find it and eat it. So when the light comes on in the morning, what the squid does is it buries itself in the sand and goes to sleep. So same squid, it just camouflaged itself. We've given it some sediments. It thinks it's morning, and it wants to hide from predators. So what you can see is it's thorough. it buries itself in this sand, and that makes it invisible to predators. And so, you get eaten by a predator once. So this squid is very serious about covering itself up with the sediment. And there it is. This is the behavior in the morning. So if I showed you this aquarium, you wouldn't know that there's a squid in there. There's actually about 10 of them in there. All right, so this is a fantastic anti-predation device that this squid uses during the daytime. It's invisible, predators can't see it, it's safe. The problem for the squid is when it comes out at night. It can't stay there all day and all night. At night, as I told you, it's got to come out to hunt. So now it's vulnerable because predators could see it at night. And so what this squid has evolved is a beautiful one-to-one symbiosis with a bioluminescent bacterium named Vibrio fischeri. And Vibrio fischeri provides the squid light that the squid uses to counter-illuminate itself. So now let me show you how that works. So here's a squid that's been turned on its back. And I hope what you can see is down at the bottom are these blue glowing parts of this squid. So what this squid has developed is a specialized light organ that's under the body or the mantle of the squid. And in this sac are these glowing bacteria, Vibrio fischeri, and they're packed in there. There's like 10 to the 12 cells per milliliter. It's like toothpaste. It's thick with bacteria. And in that light organ, those bacteria make light, and the light shines out of the bottom of the squid's body. And so what the squid does is when it emerges from the sediments at night, it's just living in sort of shallow, knee-deep water. It comes out at night to hunt, and it's got detectors that are on its back. And it can sense how much starlight and moonlight is hitting its back. And then it opens and closes a shutter over this light organ that modulates how much light comes out of the bottom, which is made by the bacteria. And so the squid matches the amount of light hitting its back with the amount of light coming out of the bottom of it so that it doesn't make a shadow. So it's actually using the light from the bacteria to counter-illuminate itself and again, make itself invisible from predators, this time at night. So it doesn't cast a shadow, so no predator can see its shadow and calculate where the squid is and eat it. So now the squid has it, both in the day, it's buried in the sand. At night, it's counter-illuminating itself by the light made by its symbiotic partner, this bacterium, Vibrio fischeri. And so the squid is now protected both during the day and during the night. Okay, so that's how it works from the squid's perspective. But now let me tell you about the bacterium's perspective. So it turns out that the bacteria only make light when they're at high cell number inside the light organ of the squid. And that is only at night. So they only make light at night. So how do the bacteria know how to do that? All right, so that's shown on this next slide. The way these Vibrio fischeri bacteria understand when to make light is by communicating with each other and only making light as a group. All right, so here's how it works. So this little oval is supposed to be my bacterial cell. So if this bacterium, this Vibrio fischeri bacterium was alone, right, it was at low cell density, so not many other cells around, making light does no good. The amount of light that one bacterium can make is incidental. So at low cell density, Vibrio fischeri makes no light. It doesn't glow. But what Vibrio fischeri does do is that it makes and releases small molecules that you can think of like hormones. We call these autoinducers, and they're drawn by these red triangles. It makes and releases these molecules, and these molecules get released outside of the cell. The world is big, the bacterium is small, these molecules diffuse away, the bacteria can't detect them. That says you're alone, don't make light. But then as the Vibrio fischeri bacteria grow and divide, as I told you, the more cells there are, since all of the cells are making the share of this extracellular molecule, this hormone molecule, the amount of the hormone or the autoinducer increases outside of the cells in step with cell number. So the more cells there are, the more of that molecule there is around the cells, because they're all participating in making it. So the molecule builds up in amount to a particular threshold concentration. And when the molecule hits that level, the bacteria detect it, and they infer from that detection event that there must be neighbors around. So in unison, all of the bacteria begin to make light together. So in fact, they only make light when they're in a group, And the way they know they're in the group is by whether or not this molecule is present. So in fact, the bacteria have no idea whether or not there are really other cells are around. They're using this molecule as a proxy for cell number, and they're responding to it, as if, because it tells them there must be other bacteria around. And we know we're right about that because if we collect up the molecule and squirt it onto low cell density cells, they will turn on light. So they're just measuring the concentration of the chemical, this communication chemical, and turning on light in response. And so the way it works in the squid light organ is that every morning when the squid goes to sleep in the sand, it's got a pump that's attached to its circadian rhythm, and it pumps out like 95% of the bacteria in that light organ. So the bacteria are in this situation. They're at low cell density, they're not making light. The squid doesn't care, it's asleep in the sand. As the day goes by, the bacteria grow and double, they make more and more and more of themselves, they're releasing the molecule, and then at night it hits the magical amount, and all the bacteria turn on light in the light organ exactly when the squid needs it. So every morning they're like this, every night they're like the left-hand side of the slide, every night they're like the right-hand side of the slide, and the squid is doing that by pumping most of them out when the sun comes up. Okay, so now you get it. You get how the squid uses these bacterial light to protect itself, and you understand how the bacteria know to only make light when they're in a group, and that's by using these chemical communication mechanisms. So we were really curious how that worked from the bacterium's point of view, and so we're molecular biologists and geneticists, and so we wanted to figure out how does the bacteria make and detect that molecule. So what we did was to discover the components that drive this behavior. Okay, so now this is supposed to be by a bacterial cell, again, my vibrio fischeri cell, and what we discovered is that there was a protein, an enzyme that made that autoinducer, the hormone molecule. And that autoinducer diffuses outside of the cell, so the more cells there are, the more of their molecule there is. So that you've already learned. But then we found that there was a partner protein, a protein that's called a receptor that actually sits on the surface of the bacterial cell. And it's got a part of the protein is on the outside and a part of the protein is on the inside of the cell. So this receptor sits there on the cell surface, just like the receptors sit on your cell surfaces. And when the autoinducer, the hormone, builds up in concentration, it slots into a groove in the receptor. And when the receptor binds that molecule, information comes into the cell to tell the bacteria to turn on the genes that make the enzymes that make light or bioluminescence. So it's actually really simple the way this works. The more bacteria there are, the more of this chemical there is outside. At a particular threshold amount, the receptor will find it and bind it, and then all of the bacteria in unison will turn on bioluminescence. And so now you get how that works. And the reason that that turns out to be particularly interesting is that after we made these discoveries in these bioluminescent bacteria, what we and lots of other scientists found is that this was not restricted to this sort of obscure marine bacteria, but this is the norm in the bacterial world. There are now thousands and thousands of examples of bacteria that have components like the ones that I've just shown you. So what we now get is that probably all bacteria have to understand times when they're alone from times when they're in groups, so they can change their behaviors based on those scenarios. And the way they do it are with chemical communication circuits. So now we have a name for this process that Anita told you. We call it quorum sensing. So the bacteria make these autoinducers. The autoinducer molecules build up outside the cells at high cell density. They lock into their partner receptors. Information comes into the cells, and all of the bacteria turn on all kinds of group behaviors that are not productive when a single bacterium carries them out alone, but that only become productive when the group carries them out in synchrony. So now we get that this is not an anomaly. This is built into probably all bacteria. I mean, all is a big word, but at least thousands and thousands of cases by now. right? So that's quorum sensing. And we also know what these molecules are that I'm depicting as these triangles. So we and other scientists have discovered the identities of these molecules. So in my cartoon, I have them as red triangles, but this white molecule is the actual chemical. That's the word that Vibrio fischeri talks with, that in my cartoons, I use the triangle symbol. And so the first one discovered, of course, was from this glow-in-the-dark bacterium Vibrio fischeri, but over time, lots of these quorum-sensing molecules have been discovered. And so on this slide, I just have a few bacteria that are quorum-sensing bacteria and the molecule that each one of these bacterium uses to communicate with. And what I hope you can see is that all of the molecules are related to one another. So the left part of the molecule is identical in every single case. And these right-hand tails, these are just carbons, are a little bit different in every single bacterium. And these tiny little differences on the right-hand sides of the molecule confer exquisite species specificities to each of these molecules. So what I mean by that is if we put the Pseudomonas molecule on Vibrio fischeri, nothing happens. And likewise, Pseudomonas is impervious to the Vibrio fischeri molecule. So each of these molecules is perfectly specific for its partner receptor. They fit like locks and keys, and there is no slop in these systems. So what we interpret that result to mean is that these molecules enable intra or within species communication. These molecules allow bacteria to talk to their siblings, but these are private, secret languages so that bacteria that are identical to one another can talk to each other and nobody else can listen in. So these are intra-species communication. This is how I count my family members and we all decide to do something. So it's private languages. Okay, so that's quorum sensing. Those are some of the molecules. And then what about the behaviors? So I told you how it works for Vibrio fischeri and bioluminescence, right? But we and lots of other scientists got interested in what are the kinds of behaviors that non-bioluminescent bacteria control with quorum sensing. And in many, many cases, globally important pathogens have been studied. And it turns out that quorum sensing controls virulence. So when bacteria are pathogens, they have to sit on surfaces, so either on your skin or on a tissue, in your lungs. They sit in surfaces and they build communities. Those are called biofilms. And so the building of the community takes lots of cells working together. Quorum sensing controls the making of the biofilm. So how bacteria establish communities, and then as a group, the bacteria release the poisons or the toxins or what we call the virulence factors, the actual biocomponents that make the host sick. And so if you think about it from the point of view of, say, the pathogen, right? The worst or stupidest thing a bacterial pathogen could do is if a couple, let's say Pseudomonas, get into me, right, and they start spitting out their toxins, right, those toxins won't have an effect because I'm huge compared to the bacteria. Moreover, our immune systems evolved to hunt down invading bacteria and get rid of them. So these virulence factors tend to be red flags for the immune system. So the better idea from the bacteria's point of view is to wait, to count your numbers, to recognize when you have the right number of bacteria, that if all of you release these virulence factors together, they can overcome the host immune system. That is how these bacteria make these productive infections. And so we know this is true. If we make mutants that don't have quorum sensing, they're not pathogenic. They have to be able to do these tricks as groups, or the tricks, virulence doesn't work. So now there's all kinds of cases of clinically relevant bacteria that must have quorum sensing to show those clinical effects, right? So this list goes on and on and on. All right, so once we got this far, that we started to understand bacteria communicate with chemicals, they talk to their siblings, they control virulence, what my gang was pretty interested in was wondering if there couldn't be more to these quorum sensing systems. And so what we were really wondering about is the way bacteria, unlike Vibrio fischeri, typically live in the world. So I told you about Vibrio fischeri. It lives in this pristine one-to-one symbiosis with this squid. But most bacteria don't live like that. They live in mixtures with hundreds or thousands of other species of bacteria. And so we were trying to understand if quorum sensing is so specific, it's these sibling, these private conversations, how can bacteria do quorum sensing when they're in mixtures? And that's shown on this slide. This picture is human skin. This is a picture of what your elbow skin would look like under a microscope. It's covered in a bacterial biofilm. It's supposed to be. And what you can see are all these different shapes and sizes. Every one of those is a different species of bacteria, the different shapes and sizes. And so what we wondered, how can quorum sensing work if there's, how do bacteria actually count accurately if there's all kinds of other species present? And so we wondered if quorum sensing couldn't be more sophisticated than we had been thinking about in our early studies. And so we wanted to explore that question. And so the strategy we decided to use was to study a relative of Vibrio fischeri. We decided to study a bacterium called Vibrio harveyi. So Vibrio harveyi is a bioluminescent marine bacterium, but it lives free living in the ocean. And so it lives and encounters all kinds of other bacteria, right? But it has quorum sensing. It turns on light at high cell density. So we thought this would be a good system to study. And so the reason I'm showing you this slide is to try to give you an idea of some of the things that we do in the lab. So what you're looking at is a flask and some Petri plate and some ridiculous graduate student propaganda, right? And these are all pictures of Vibrio harveyi. And all we did to take this picture was just turn the lights off in the room. This is what we actually see. We actually took the picture from the light, the bioluminescence that the bacteria are making. Right, so they are making that light because they're in groups and they are doing quorum sensing. And what was so fantastic about Vibrio harveyi and earlier Vibrio fischeri is that the output of quorum sensing, what the bacteria do in response to quorum sensing, is make visible light. Right, and so I always wonder, why did this get missed for 480 years? Right, but of course bacteria are invisible. Right, so if they're doing something alone or something as a group, how would you know it? How would a scientist know it? You can't see the bacteria, you can't see their behaviors. But what was so amazing about bioluminescence is it made the invisible world of the bacteria visible to us, the scientists. We could just see when they're doing quorum sensing because they make light. And so what we could do then is make mutants, just randomly mutagenize Vibrio harveyi and look for bacteria that couldn't make light when they should. And by doing that, we just looked for mutants that were dark. We could find those mutants and find the components of the Vibrio harveyi system and earlier the Vibrio fischeri system. Okay, so we're studying this wild free-living bacterium. We're using this bioluminescence to track quorum sensing. So what did we find? So when we studied the Vibrio harveyi system, we found something similar to what we had found in Vibrio fischeri. There was a protein and an enzyme that made an autoinducer and then the partner receptor that detected it. And I actually showed you this molecule. It looks like similar to the others. I showed you that on an earlier slide. But to our surprise, when we got rid of this system, what we found was there was another quorum sensing system in this bacterium. So there was a second protein that made a second autoinducer molecule, and it also had its partner receptor. So there were two quorum sensing systems feeding in to tell the bacteria when to turn on bioluminescence and all kinds of other genes. So there were two circuits. And so when we got those results, we started to wonder, what is the point of these two molecules? And what we thought is if these two molecules don't encode different pieces of information, having two systems isn't better than one. So we knew what this system was for. It's for intra-species communication. I've already shown you that. So we wondered, what is the role of this second system? And so to find the answer out, what we did was we purified this molecule, and then we just scanned all kinds of bacteria and asked whether or not they made it. And what we found was that every bacterium we tested, every bacterium, no matter where it came from, made that second molecule, and every bacterium made the identical molecule. So this is what we call autoinducer 2, because it's the second one we discovered. these are carbons. It's a 5-carbon molecule with lots of oxygens on it. That appeals to me. That's not the important point of it. The important point is that every bacterium makes that exact molecule. So this is really different than the first system I told you about, where every single species makes a slightly different molecule than every other species. In this case, the molecule is generic. They're all talking with the same language. So we think this molecule enables interspecies or cross-species communication. This is a universal communication molecule. It's like the bacterial trade language or the bacterial Esperanto. So now what we think is that bacteria must be multilingual, at least bilingual. They have some way to know self, and they use this second molecule to know other. And so what that means is that they can talk across species. And so the computation that we've shown that bacteria do is the following. Bacteria are out living their lives in their whatever lifestyle they have, and they want to know, the first thing they want to know is, am I alone or am I in a group? And so they're monitoring their environment for quorum sensing molecules. And based on what they find out, they decide they're alone or they're in groups, and they begin to turn on genes, all kinds of genes that underpin group behaviors. But then the more sophisticated computation the bacteria do is they measure the ratio of these two signals, because this red one means self, the pink one means other. And then what they do is they tailor what genes they turn on based on whether they and their kin are in the majority or whether somebody else, the enemy, is in the majority. So they can tell self from other, and they can actually react to that. They can decide, is it me and my siblings that are winning or losing? And based on that, they turn on genes that are good for themselves or harmful to somebody else. So they figure out if they're winning or losing, and they behave appropriately. So they know self from other, which seems like a very sophisticated trait, but we think that evolved billions of years ago in the bacteria. Okay, so that is quorum sensing. You know a lot about it now. But what I promised I would tell you is, based on what we learned, could we then do something more practical? Like, now we get the bacteria communicate, we know something about the molecules, and we know that these traits are really important in medicine. So what we wondered is, could we interfere with quorum sensing to make new kinds of antibiotics? And probably definitely know microbes are a problem right now, bacteria, as well, and that we're out of our arsenal of antibiotics, that there's a global problem with antibiotic resistance. And so the question is whether or not we can think of new ways to combat harmful bacteria. So what my gang would like to do is to make bacteria that can't talk or can't hear, because they wouldn't understand they're in a group and they wouldn't turn on their virulence traits. And so there's sort of two strategies. One is, if you want something very specific, like your physician knows you have a particular disease caused by a particular bacterium, you'd like to interfere exclusively with the intra-species communication circuit, so you could just have a precise medicine that combats a particular pathogen. Alternatively, sometimes people get sick, your physician doesn't know what you have, and so you want to treat broadly, you want a broad spectrum antibiotic. In that case, perhaps we'd like to interfere with this generic system, because maybe you could shut down quorum sensing in all kinds of bacteria while your physician tries to narrow down what you have and give you a more appropriate therapeutic, right? So we are working on making both of those kinds of anti-quorum sensing molecules. The logic for how you get there is the same. So tonight, because that's our newest results, I'm going to show you about our efforts to shut down intra-species communication in hopes of making new medicines. So we are working, excuse me, in a notorious pathogen that I've said the name before, which is called Pseudomonas originosa. So Pseudomonas is a pathogen that just lives in the dirt, it lives outside. You and I encounter it every day, and it's harmless unless you are immune compromised, or you have the disease called cystic fibrosis, or you are a victim of burns. So for example, people who have the disease cystic fibrosis, you might know, they have a genetic mutation in their lungs. And so they can't clear their lungs of bacteria normally, the way most people that don't have CF can. So what happens is, typically when a person is in his or her teens, they become permanently colonized by Pseudomonas originosa. We don't know why, but people that have cystic fibrosis die of a Pseudomonas originosa infection. Same is true for burn victims, for immune compromised people, and also Pseudomonas originosa is one of these terrible pathogens that's sort of running around in hospitals. You know, you go to the hospital to get your gallbladder removed and you get a Pseudomonas infection. So it's a real problem both for this demographic of people who are immune compromised or have cystic fibrosis, but also it's just a general, increasingly general problem in hospitals. The reason Pseudomonas is so deadly is because it has quorum sensing. It is a notoriously sticky bacterium. It loves to make biofilms. And you'll remember that I told you biofilms are how bacteria sit on surfaces. So these could be biotic surfaces like the lungs of a CF patient, or abiotic surfaces like all the machines and the surfaces in hospitals. So Pseudomonas loves to stick to surfaces in these communities called biofilms. That's a group behavior. Quorum sensing controls the making of biofilm communities. And then as a group, Pseudomonas has 50 or 60 virulence factors that it secretes. And it's those factors that damage a person's lung tissue, that are poisonous to people who are immune compromised, right? Those are the proteins, the poisons that this pathogen uses that make it deadly. So both of those traits, the ability to make the community that's together and then to produce those virulence factors, all of those traits are controlled by quorum sensing. So if Pseudomonas doesn't have quorum sensing, it cannot make a biofilm and it does not produce virulence factors. So we thought because this pathogen is so deadly and we knew a lot about its quorum sensing system and its virulence behaviors, this would be a really good pathogen to try to test whether there's merit to the idea of making anti-quorum sensing therapeutics. All right, so how do we do it? So this is the Pseudomonas autoinducer. I've shown you this molecule. This is the natural molecule that Pseudomonas uses to control these group behaviors. So we knew that molecule. And so what we did in the lab was we made an inhibitor. And so this is the natural molecule. The right-hand side is the synthetic molecule. And so what you can see is that this part, hopefully everybody can see my cursor, this part of our synthetic molecule is almost identical to the Pseudomonas molecule. There's an oxygen here, there's a sulfur here, right? So the first part of our molecule looks like the real thing, but on the right-hand side of our inhibitor, there's a big group. It's sort of like a bump. And so what happens is when this, remember, the real molecule is supposed to slot into that receptor, like I told you, and turn on quorum sensing. When our molecule slots into the receptor, it gets stuck there. It jams the receptor. It doesn't turn on quorum sensing, but because the receptor is filled, that slot is filled with our inhibitor, the real molecule can't get in. So this molecule inhibits reception of the native autoinducer, right? So this is called an inhibitor or an antagonist. And we made it based on our knowledge of the real Pseudomonas autoinducer. Okay, so the idea is we're going to jam the receptor, shut down quorum sensing, and then Pseudomonas shouldn't be a pathogen. Okay, so does it work? All right, so now you know our molecule, so can we actually do this? I mean, you know, make this idea work. All right, so what you're going to remember that I told you is that the first step of the infection is that Pseudomonas and pathogens, they have to sit on a surface and make a biofilm. So that's shown on this slide. This is a Petri plate. And what we did was we just put a little drop, here where my cursor is, we put a little drop of Pseudomonas on that Petri plate. And then we let the Pseudomonas grow, and what you can see is it spreads out, it's covering the plate in this beautiful pattern. This is a biofilm. So Pseudomonas just grows on the surface of that Petri plate and makes this biofilm. If we do the same experiment, but into the plate, we put that inhibitor, that anti-quorum sensing molecule, and then we put the drop of Pseudomonas in the middle, what you can see is that Pseudomonas can't move out, it can't make the biofilm, the inhibitor stops it from that first step. Okay, so that looked pretty promising to us. The anti-quorum sensing molecule prevents biofilm formation. All right, so what about step 2? Beyond the biofilm, right, Pseudomonas secretes these virulence factors, these toxins and these poisons that I told you about. So we wanted to test whether our inhibitor could shut that down as well. So that's on this slide. So we have an animal model that allows us to test Pseudomonas infection. So what we're doing is measuring how many of the animals are alive over time of the infection. So of course, if we don't, this is a control, if we don't infect the animal with Pseudomonas, all the animals are alive. Okay, so that better be true, right? And then if we infect the animal with Pseudomonas, the animal succumbs. So in 24 or so hours, nearly all of the animals have died. That's because Pseudomonas has made a biofilm and it's secreted these virulence factors, and those kill the animal. All right, so those two experiments, we knew how those were going to work. And so then the real test is if we do this second experiment in yellow, we give Pseudomonas and we also provide our anti-quorum sensing molecule at the same time, what happens? And what you can see is that the animals live. So indeed, if we give this anti-quorum sensing molecule, it shuts down quorum sensing, and Pseudomonas is not a pathogen. So we're pretty pepped up about that experiment. This is not a medicine yet. This is, what's called a lead molecule. It's not particularly potent. We have not shown it's safe. We need now that we have merit to this idea, now what we're trying to do is to do the medicinal chemistry to make the molecule go where we want it to go. It has to last a certain amount of time. It can't go where we don't want it to go. It can't have harmful side effects, right? And it needs to be much, much more potent. Right now, you'd have to take a pill that's, this big, right? So anyway, but we needed to get merit for the idea. And so we think we're there now. And so we're trying to do this with this molecule and many others. And so we think that there could be medicines made from shutting down quorum sensing. And so we're working in that direction. And so that's the stories that I wanted to tell you tonight. Let me finish by just doing a little summary, if I may. So the first thing that I told you is that bacteria talk to each other. Their language is chemical. They use chemicals as their words, and those chemicals allow them to count and understand when they're alone and when they're in groups. What we think that means is that bacteria are multicellular, right? So, we all evolved from bacteria, right? And it was thought for a long time that bacteria are sort of the asocial part of the evolutionary tree. What we would argue is that quorum sensing allows bacteria to be multicellular. And so perhaps some of the discoveries we're making about the system will help our colleagues understand higher organisms and how multicellularity works and how multicellularity evolved. With the two molecules I showed you, what I hope you've learned is that bacteria can distinguish self from other. And again, that's something that always happens in higher organisms. It's not like your heart cells and your kidney cells get all mixed up every morning, or every day. That's because they each have their own set of chemicals that they use that tell them what are their jobs and what traits should they carry out. And so again, we think that evolved billions of years ago, and that the skeletons of those systems exist in bacteria. And by learning about bacteria, we'll be able, hopefully, to have insight into higher organisms as well. And so then in the applied part of this work, of course, what I've shown you tonight is that we're trying to develop strategies to impede harmful bacteria in an effort to make new medicines to make animals, plants, and humans more healthy. But to go back to the very beginning of my talk, you're going to remember that you have this magical microbiome that lives in and on you, and every higher organism does, right? And those bacteria are keeping you healthy, and we know they have quorum sensing. So besides making anti-quorum sensing molecules to stop harmful bacteria, we're also trying to make pro-quorum sensing molecules to make beneficial bacteria even better. And so perhaps, and we don't know yet, the real way forward in medicine is to actually improve the chit-chat of these bacteria that naturally live with us and let them do the fighting against these pathogens, right? So what we know now is that our microbiomes are working hand in hand with us to fend off these invaders, right? And so maybe we can enhance the microbiome quorum sensing, and they'll do the work. They'll be sort of natural medicines in us, right, that help us to stay healthy when we're invaded by pathogens. And then finally, the last thing is to finish with a confession, which is the bacteria already discovered all of this. So once we started to get this idea, like, maybe we could manipulate quorum sensing, on purpose, we thought, well, wait a minute, the bacteria have a 4 billion year head start. You know, they're out there duking it out and living in these competitive environments. And so we and lots of other scientists have now started to go out to the natural world and find out the strategies bacteria use to compete against one another, because those strategies that exist in the natural world have been tried and tested over evolutionary time, and they're probably the best strategies that there are. And so we should bring them back into the lab and tinker with those. And so sure enough, if you go out and look in natural bacterial communities, they make anti-quorum sensing molecules, they eat each other's quorum sensing molecules, they cut each other's molecules in half. So one guy's trying to talk and the guy next to it is shutting it down. And so all of those strategies exist in the natural world, and we're trying to use those as inspiration now for our studies and maybe make them a little bit more refined, but this might be the real way forward in the applied part of the studies. Okay, so that's where we are. And let me finish by showing you my gang. Obviously, this was taken before COVID. This is my lab group and our families out on my porch during happier times. And I just want to make sure that everybody knows that I have the luckiest life in the world, because I work with all of these budding scientists, people who are trying to become scientists, that have these creative and wonderful ideas. They're fearless, and they are the ones who change the way we think about the natural world. And so I'm really lucky, because these people want to come and work on this quirky project. But I just want to make sure that you know that every discovery that I told you about during my talk tonight was made by somebody's brain and somebody's hands, not mine, who are in this picture. And they're really pushing the forefronts forward, right? And I think, you know, scientists now are in the news, and maybe people, you know, are really starting to believe that we're going to come to the rescue. We've been doing that for a long time. And I think that these faces show you that the people that do it, and they just change the way we think about life on Earth and the possibilities for humanity. And anyway, so that's my gang. And I guess I will stop there and take questions if there are any. And I just want to thank the Wistar again for this fantastic, meaningful prize. And I thank everybody for listening and learning something about what my lab does down the street from you guys. So thanks.
Bonnie, thank you so much. That was so, it's, the work is so elegant. How you did it is amazing. And how, I think, you know, you're one of the best examples of how basic research is so critical to understanding how we get to medicine, because you understand the whole process, and you're actually adding insight across not only infectious disease, but, you know, now that we know that the immune system and the gut and the bacteria all speak to each other, this is going to change mental health. It's going to change everything. So thank you so much. You're such a bright star, and I'm already getting rave reviews in the background of your presentation. So thank you. Okay, so I stopped gushing. Let me ask a couple of questions. So one of them is, so can you tell us a little bit about the research going on between the human gut flora and the immune system modulation, and how this may be correlated?
Yeah, absolutely. That's a fantastic question. So I spend my 45 minutes talking about how bacteria talk to bacteria, right? But now, you know, and then I showed you it's intraspecies and interspecies. So then somebody in this gang said, well, what about interkingdom communication? So sure enough, this is a dialogue. It's not a monologue. And what we're now, and we know about the microbiome. We know we're, you know, got all these bacteria on us. And so very recently, my gang showed that in fact, the host, you know, the human cells are listening in, and they actually make their own autoinducers. So we think that they are driving this conversation, right? So it's going to be between kingdoms, where the bacterial molecules modulate what, for example, your gut cells, your immune cells do, and likewise, your cells talk back to these bacteria, and presumably the conversation should matter whether it's a hostile bacterium or a beneficial bacterium, right? But it's really clear now that this communication spans kingdom boundaries, and that is the horizon for us. Even more complicated to figure out, but we're on it, right? And our newest molecules come from human intestinal cells. So yeah, whoever asked that should come work in the lab, because that is the forefront for us, right? To really up the complexity and understand how this works in real systems that have lots of different cells in them.
That's crazy. Okay. So, so one other question is, if you impede quorum sensing, could you also hurt some of the communication between good bacteria? And is there a concern of overlap between those, even though you've got the intra versus interspecies molecules?
Right. Also a great question. So, if we do the interspecies molecule, you are right. We will shut down the chit chat of everybody, let's say in your gut. But we are banking. With the intra-species molecules, you might be able to selectively shut down somebody. Okay, but for sure the interspecies, everybody's going to be like not chit-chatting correctly, and we are banking on the almost 100 years of traditional antibiotics, right, that work, right? So when you take an antibiotic, you don't just kill the one bad bacterium, right? That's why you get a stomach ache. Women get a yeast infection, is because you kind of carpet bomb all the microbiome bacteria, good and bad. You get rid of the pathogen, right? And what the miracle of is that antibiotics ever worked, right? Because what is so amazing, right? And they do, right? One of the greatest public health things ever, right? What's amazing is that in our microbiomes, we have what are called persister cells that aren't harmed by antibiotics. And so after you take away the antibiotic treatment, these persister cells regrow and your microbiome comes back. That's why you survive it. So if we can really make it, so that's really tough, you know, and you get these stomach ache and side effects, right? But it works. And so if we can actually do the anti-quorum sensing therapies, it's a much softer, you know, we aren't actually killing the microbiome. We're just making everybody be quiet for a little while, give your immune system a chance to get rid of the pathogen, which is what it does when it doesn't express its virulence factors. And then your microbiome will still be there, you know, when we remove the therapy. I mean, that is the fantasy. We have to show that that is true. But we actually think we're going to be better off, right, with these kind of behavior modification strategies rather than killing strategies, just for that reason.
Yeah, I mean, it reminds me so much of the difference between chemotherapy versus personalized medicine. And, you know, while thank God for chemotherapy, if we could not kill every cell, that would be a huge benefit. So it's fascinating. Okay.
The exact logic. Yep.
Yeah. Okay. Another question. Disease interruption and mitigation is important, but can your research also inform how we might get microbes to behave in beneficial ways to humans?
Right. So I hinted a little bit to that at the end, like that perhaps we could make pro-quorum sensing molecules to enhance the microbiome. And what we know is that your immune system and your microbiome are working hand in hand to fend off predators, right? Invading bacteria. So if we could make the microbiome better, that is one way. And then the other thing, which I didn't allude to tonight, is that we use bacteria as machines, as factories. You know, we use them to make products for agriculture, for medicine, for industry, right? And if we could actually, you know, they're just metabolic, you know, warehouses, you know, that we're trying to mine, and people and industry uses them all the time, you know, like bioremediation, you know. And so if we could actually make these pro-quorum sensing molecules, right, to both for the microbiome and health purposes, but all these applied purposes where we already use bacteria, right, to make us stuff, I think there's so many applications, right, of either stopping, you know, bacteria that we don't want somewhere, or helping bacteria that we already use or that we can imagine for new uses in the 21st century. Right, people in my field have our eye on that, right?
Yeah, it's a whole nother manufacturing world. I told everybody you'd never think of bacteria the same way after this presentation. Okay, last 2 minutes, because we do love being punctual. Bonnie, what keeps you up at night? And on the flip side, what gives you hope?
Yeah, okay, so the people in that picture give me hope. I'll start to think like, so these young people that, you know, they're typically young, that want to go into science, that they're fearless, they don't believe anything they've read, they believe that their lives matter and that they can make a contribution, right? Like they're like, I'm getting old and dusty, and they're always just as energetic, they're always just as out there, and like there's something so special about the way the United States does science, right? It's inefficient. They come and go. Nobody stays with me more than 4 or 5 years, but it's always refreshing itself. And that always gives me hope, right? So for sure, and and that's true in all the labs in the United States, right? So I think you can have incredible hope, right? Even like right now, these scientists made a vaccine in a year, right? For a virus that was never known, right? Like, so, like, I think that is an exemplar of what goes on in these labs all the time. What keeps me up at night? Antibiotic-resistant bacteria and viruses, microbes,
Yeah.
right? So microbes. So what really keeps me up at night is that I think even in the midst of COVID, right? Microbes are before COVID, after COVID. Microbes are what kill most people on Earth. They die of infectious diseases, right? Not boutique diseases like we die of. Most people don't live long enough to get cancer, right? Or to have heart disease, right? And I think that COVID's a kind of wake-up call, but I think that the world does not appreciate how, now they do a little bit, how incredibly serious and how vulnerable we are to pathogenic microbes. And it's sort of passé to be working on antibiotics. And that, to me, that kind of mindset has got to change. And maybe if there is a silver lining, in my field from COVID, it's that people understand we are all connected to each other. We are all vulnerable. These organisms are invisible, right? And that we've got to study microbes and we have to make arsenals of therapeutics. That keeps me. I'm worried that we're going to have a 2-second memory once we, get past this terrible crisis.
Totally agree. Okay, and on that note, Bonnie, congratulations from all of us at Wistar. This was just a joy, and I was looking forward to it forever for the right reasons, because you, as always, were amazing. So thank you from all of us, and we look forward to seeing your rat high on your shelf with all of your Zoom calls. And there we go. And we look forward to seeing you here at Wistar when we all open back up again.
It would mean happier times.
Thank you. Well, thank you to everybody for joining us tonight, and we'll look forward to seeing you at our next Women in Science presentation, and good night.